My name is Caitlin Hester. It's November 10th, 2019, and this is my Unit 2 Case Connections video presentation. For the first one I answered, it was case 1, and it talks about the regulation of pyruvate kinase in the cell, and it's important to remember there are 4 different isomers related to pyruvate kinase. You have ones found in the liver, red blood cells, and then skeletal tissues, and then others in stem cells and the rapidly proliferating cells. I want to first talk about the difference in structure. So pyruvate kinase is a tetramer, and it's made of 4 identical subunits, that have 3 domains, A, B, and C. C is where you would bind the PEP and the ADP, and you'd have that phosphoryl group transfer. And PKM2, L, and R are all activated allosterically. But PKM1 is non-allosteric, and it just kind of resides in a constant active state. And it's a very highly specialized conformation, and it is in the high-performing tissues that require a lot of oxygen production. So what regulates pyruvate kinase is mostly fructose-1 bisphosphate is the major activator. And then you also have ions, and you have monovalent and divalent ions that kind of synergistically work together. One of the most important ones in this is potassium, and this interacts with the phosphoryl oxygen of PEP. And what they do in the divalent ions would be like magnesium or manganese, and these ions kind of work to facilitate an interaction of pyruvate kinase and the PEP. And then specific to PKM2, it's called succinylamino imidazyl carboxamide ribose 5-phosphate, or SICAR. And this doesn't necessarily activate, but it helps to allow cancer cells to survive and grow in low glucose conditions. So it kind of augments the work of PK. Serine increases in serine concentration, increased PKM2 activity levels. Insulin stimulates pyruvate kinase expression. And then in terms of inhibitors, ATP is an allosteric inhibitor. Basically, increases in ATP is the way of signaling back, saying we already have enough product, we don't need you to keep going. And then alanine reduces the affinity for PEP, the enzyme pyruvate kinase's affinity for PEP, and it also works to make the tetramer more stable in the T state, or the inactive state. And it also works to tell PK there's already enough building blocks made and don't make any more. Thyroid hormone T3 weakly inhibits pyruvate kinase, but if there's enough fructose bisphosphate, then it takes care of that. Glucagon found in starvation states inhibits PK expression in ancillary tissues. Basically, it works on the PKL, the pyruvate kinase with the L isomer, to say, don't use up all this glucose here in the liver. We want to save it for tissues like the brain, skeletal, and heart tissues that need the glucose in this starvation state. So, and for part 2, it's asking the question, how come PKM2 is found in this low activity state and needs that allosteric activation? Why aren't cancer cells just using like PKM1, which is always active? But looking at the Warburg effect, so how cancer cells work is the rate of glucose uptake in these tumors or rapidly proliferating cells dramatically increases, and it converts glucose to lactate, despite fully functioning despite fully functioning mitochondria and the ability to participate in oxidative phosphorylation. The cancer cells are just going through glycolysis and making lactate. So cells that express PKM2 use a lot less oxygen. They don't need that PKM1 where it requires a lot of oxygen to work, essentially. So that's part of the reason why PKM2 is preferable. And because PKM2 is more inefficient at making ATP, it compensates by increasing glucose transporters and glycolytic enzymes. So it brings in all of the glucose and glycolytic enzymes that cancer cells need because they are taking in so much glucose in their way of metabolism. And then PKM gene is expressed through splicing of pre-messenger mRNA, and so the oncoprotein MYC basically inhibits the expression of PKM1 and leads to an overexpression of PKM2, and that's because PKM2 helps to create a more favorable environment for cancer cells and rapidly proliferating cells. And then PKM1 also is needed for the epidermal growth factor receptor, and this is a key component in tumor cell growth. In Nofricase 2, it talks about the young child that presents with for an annual visit, and they have really low serum insulin levels. And then after a glucose load, they still have very low serum insulin levels compared to a control group. And so basically what this information suggests about the biochemical basis is that the patient has an insulin deficiency. The body is not making enough insulin, and so that leads to the diagnosis of type 1 diabetes, which is a lack of insulin production. Whereas in type 2, you would still have the insulin, but there's a problem with sensitivity, and so that's not the boy's issue. So then if you were looking at the fasted glucose levels, the body is in a hyperglycemic state constantly. So the fasted blood glucose levels will still be above the normal range because no insulin has been made, or not enough insulin has been made to take that serum, that glucose in the blood into the beta cells and start to process it. So in glycolysis, when the body is in a hypermetabolic state, you would see enzymes such as hexokinase and phosphofructokinase playing an important role in regulating glycolysis. You've basically given the body all of this glucose to work with, and then the enzymes hexokinase and phosphofructokinase come out to start getting the ball rolling on glycolysis. So phosphofructokinase is an important element in the glycolytic pathway. It works to help create fructose 1,6-bisphosphate through phosphorylation, and we all know as glycolysis continues, that starts to lead us down towards what we talked about in the first case of getting to the point of the end products of glycolysis with with pyruvate kinase and getting that pyruvate in the lactic acid. And then hexokinase, this catalyzes the first step in metabolizing glucose through phosphorylation. And so those are the two enzymes that would be likely present in the current state of hyperglycemia for this individual. And attached to my paper are all the references I have used to answer these questions. Thank you.